Okay, I'm going to introduce our next speaker, Asif Ghazanfar. Do I say your last name right?
Sure.
Okay, good. Okay, I used to be introduced as Lola Grubenster. I'm not joking. Asif is a professor at Princeton University in the Department of Psychology. He's done a lot of really nice work with non-human primates, primate behavior, and behavior-brain relationships, direct relationships, which are really beautiful. Vocal cooperation. We've known each other for a long time. Vocal cooperation, the importance of facial expression. He's going to talk about some of that stuff today. He has many, many publications. He told me to just say his name. He also has a number of prizes, including a Krieg. You don't have a?
Not yet.
Oh. Well, you got to get a man right on that. Anyway, you know what? I'm going to introduce you. It's Asif. You guys will get a chance to talk to him after his talk and during the talk. Interrupt, but not too much. Asif, thanks.
Yeah. As long as I still get my 2.5 hours.
You know what? It gets closer to lunch and people get ornery.
Yeah, I know. You guys can just start walking away. I'll get the message. So thank you, Leah, for that wonderful introduction, but especially for inviting me to this fantastic workshop and location. So I was trained as a neurophysiologist, but I quickly became somewhat disillusioned with what I was doing, which was I was studying neurophysiology in anesthetized rats, and I always wanted to look at the neurophysiology and then make a claim about what they were doing with that neurophysiology when they were behaving. And that was just a gap too far. So I quickly realized that I wanted to study behavior first, characterize the behavior, and then try to figure out, okay, what's the neural circuitry underlying that particular behavior. And that's kind of for this new set of data that I'm going to talk about today, which is on vocal cooperation, that's the direction of the research, is to always start with behavior. And I want to start off with acknowledging the many super smart people that I get to work with. And that's, when I call them super smart, I actually mean that, but I'm also going to actually use that as an excuse during my talk, because sometimes I don't understand what they're telling me. So there'll be slides there, you're going to ask a question, I'm going to be like, I wish that guy was here right now to answer that question. But I think I get the general gist of what's going on in my lab, and that's what I'm going to present to you today. But let me start off by talking about, well, why am I interested in vocal communication at all? I'm interested in, there's natural biomedical reasons, such as, there's a variety of communication disorders, but I really got interested in kind of in a more basic way that's better summarized by a real luminary in the field of evolutionary biology, which is this guy named Thomas Huxley. Does anybody know who Thomas Huxley is? That's that guy. So Thomas Huxley was known as Darwin's bulldog. So he was a good friend of Darwin's, and he was known as Darwin's bulldog because when Darwin, put forth his theory of evolution by natural selection, he knew that it had implications for human evolution, and he didn't want to have to deal with the public outcry of his theory. So he kind of went to his little house in the farmlands, and his friend Thomas Huxley was more than happy to argue the case for human evolution by natural selection. And he said in his book, he basically said, the question of questions for mankind, the problem which underlies all others and is more deeply interesting than any others, is the ascertainment of the place in which man occupies in nature and of his relations to the universe of things. So he's basically saying, you know what, one of the most interesting things that we could study is what makes us unique, what makes us special in the animal kingdom. And then he actually formulated what he thinks specifically makes us special. He said, believing as I do that the possession of articulate speech is the grand distinctive character of man, I find it very easy to comprehend that some inconspicuous structural differences may have been the primary cause of the immeasurable and practically infinite divergence of the human form from the simian strip. So what is he saying there? He's saying basically, you know, what really makes us unique is the way we communicate with each other. And then he's saying additionally that maybe the way that we communicate with each other, that special thing that we do, is really the result of a minor tweak in circuitry. Okay, and this is, you know, that sums up the things, some of the reasons why I'm interested in studying the evolution of human communication. And so for my talk, I'm going to take two paths that you're already familiar with from yesterday, and they're summed up first by nothing in biology makes sense except in the light of evolution. So there's a broader evolutionary question, and this question, or this idea, really has as its foundation the idea that nothing in biology makes sense except in light of development. Let me know when I can fill in the blanks. Sure. Okay, so one of the perspectives, that the real kind of foundational idea in the lab when we try to tackle anything is that we think of communication as an emergent property. We don't think of the brain as working in isolation and somehow it just sends out commands. We basically think of communication as emergent property between the body, morphology, anatomy, the brain, as well as the context. And we think of this emergent property as something that changes through time. And there's three different time scales that you can think of. There's real time, so two animals that are communicating, or two humans that are communicating. There's developmental time, and of course there's evolutionary time. And I'll try to touch upon those three time scales as we move along. So in the lab, we have many model systems for communication that each have their own particular strengths. Of course, much of what we do is with reference to humans. So there's an aspect of the lab that's trying to understand, you know, what are, you know, features of human communication, particularly speech as opposed to language. A lot of the work that we've done in the past has been done on macaque monkeys and trying to understand, well, how do we integrate dynamic facial motion with the things that we hear to help us actually hear better. There's recent work that we're doing in kind of semi-field conditions to understand the role of vocalization in assembling groups of animals together, and we do that work on lemurs. What I'm going to talk to you about today focuses almost exclusively on a New World monkey species called the marmoset monkey, and I'll explain why we choose a marmoset monkey. But the marmoset monkey is basically, it's not so impressive because it's about this big, and it squeaks a lot, but those squeaks are very important to us. Okay, so I started off by mentioning cooperation and vocal cooperation. What do I exactly mean by that? One way to think of it is thinking of conversations as a form of vocal cooperation. And what do we do in conversations? We take turn. If we were take turn, so one person speaks, and if it's a polite conversation, the next person waits their turn and then says something, and that's the way that we can actually exchange information, exchange ideas, and perhaps I would argue actually evolve more sophisticated forms of communication. If you don't have this form of cooperation from the outset, it would be very difficult to get more sophisticated forms of communication to evolve or develop. So I think of this type of vocal cooperation as quite foundational. And if you look at across different languages, different cultures, there's two kind of basic features that seem to be universal, which is you try not to overlap with other people. Again, we're talking about polite conversation. Everyone's going to talk about, oh, well, in faculty meeting, I do this. And we're talking about polite conversation. There's usually no overlap, and there tends to be a little bit of gap. And those are universal features of turn-taking during conversations. Okay? And that's a little, so I'm going to start from that as the foundation. And I'm going to provide you two origin stories. One origin story has to do with development, and the other origin story has to do with evolution. And I'm going to start with just an overview, a very, very brief overview of what the developmental origin story looks like, and which is this kind of communication starts very, very, very early. Okay, it starts in utero. How does it start in utero? Well, it's in utero that you actually start producing cries. Okay, so it's almost like there's spontaneous activity the nervous system is generating. Some of that spontaneous activity generates cry expressions. As soon as you are born, you produce those cries, and those cries elicit contingent responses from caregivers. Okay? And as you grow up in the first couple of months, sounds, seemingly random sounds that an infant makes elicit contingent responses from the mom or the father or some other caregiver, and those responses have structure. So early on, and I saw this with my own kids or watching my wife interact with our own daughters, early on the infant will just say nonsense, and my wife will basically structure that as a conversation. So the baby says nonsense, and my wife waits a little bit and says her own nonsense. Okay? And then it goes back.
You're being recorded.
Just with the kid I'm talking about. And so it starts very early on that you have this kind of a conversational exchange, and I would argue that that basically sets up this, and pretty soon, I think it's a little after, I'm going to get the date wrong, but later on, basically, the kid can start detecting, oh, I'm supposed to wait a bit, and that gives time for my parent or this other person to give a response. So they're seeding the floor, basically. And it's these interactions that's been argued basically set up the scaffold to allow a human, basically, to have conversations with anybody that they come across. Okay? So that's in a shorthand what the developmental origin story is. Yeah, Paul?
Question. So, in utero, yeah, you've been hiding it, Tony. So, in utero, when these vocalizations are being exercised, you already described it as communication. Is that, just for clarification?
I wouldn't. I think of this as the kind of the practice for this. So there's no actual communication going on, of course. I mean, in one sense, you know, when the infant moves, the mom can feel it and she can adjust her behavior accordingly. But I don't know if she can. Or, you know, I know of no evidence that the infant is crying and am I doing this?
No, but it raises a bit the question how we then define communication. Right, so does it require, in your opinion, a receiver of the message, or it can be done without that receiver? Because in utero, that might not be so apparent.
Yeah, so in the way I'm going to define communication, which I may change as the talk goes on, it requires a...
So we might agree
it requires a recipient, right? It's communication is being able to modulate the behavior of another individual. And that's why I mentioned that, you know, maybe it is that the mother can detect such movements and may change. So that's, very briefly, the developmental origins. But now I want to address first in this presentation, what are the evolutionary origins? Usually when you ask this kind of a behavior question or even a neural question, and you want to ask, okay, well, how did this evolve? The only methods that we have available to us that you've probably already heard about is the comparative method, because the brain doesn't fossilize and behavior doesn't fossilize. So the idea is you take a human behavior and then you look across this tree and find out, well, which other primate species share that behavior, and then you can say, okay, well, the last common ancestor of those two species also probably had that behavior, okay? So in the case of turn-taking or vocal cooperation, we would normally think of, okay, well, this is us over here. It should be a good probability that chimpanzees should show some preliminaries of this kind of vocal cooperation. But it turns out they don't at all, okay? So chimps versus humans. Chimps don't seem to use vocalizations that much, period. There are very few kind of studies that characterize the vocal repertoire of chimps, and the attitude in the field is generally that they don't have much to say or they don't say much, okay? The caveat to that is actually since they're they're very difficult to actually study, and they may be doing a lot of, especially in the wild, they may be exchanging vocalizations in close range, but it doesn't seem like that's the case. So they don't vocalize that much. They don't seem to be very cooperative with each other. There's some exceptions, like maybe during hunts, they seem to have some organizational abilities to facilitate their hunting of monkeys. But otherwise, and people have done studies in captivities, chimps don't really seem to want to help other chimps out or facilitate another chimp's ability to achieve some goal. And that's kind of related to this third thing where chimps aren't pro-social. So what I mean by pro-social, what I mean by pro-social is to help somebody, help another individual that's not necessarily related to you. Okay? We do that all the time. Chimps don't seem to do that at all, although there's some evidence that bonobos do that a little bit. Okay? And so we have this kind of picture about vocalizations and the lack of cooperation. And then, so we're forced to, or people are forced to come up with like, well, how do we get from, since chimps are most closely related to humans, how do we get human-type vocal communication from chimps? And what the result of this, all this cogitation about how you get chimps to become like humans has resulted in various forms of what's known as gestural origins hypotheses for human communication.
But Asif, one question. Can't you argue that in, let's say, grooming behaviors, that there is turn-taking?
It is turn-taking, but sometimes, but not necessarily the reciprocal. So dominant animals tend to groom subordinate animals, but not the other way around. Whereas we would not think of that in terms of human.
But it would be a form of turn-taking that adheres to certain hierarchical structures, but still is turn-taking in some way, you could argue.
Fair enough. But it's not vocal turn-taking.
You got me there.
That's right. Well, I mean, that's an important distinction, because what that leads to is the following: is people come up with ideas about, well, how do you get to our kind of sophisticated form of communicating with each other with our voices from these kind of manual type behaviors. So what people basically suggest, many people suggest this, so people like Tomasello, all the mirror neuron guys support this idea that basically the ancestral human was chimpanzee-like in producing sophisticated manual gestures, and they learned to cooperate somehow using basically some sort of protoform of sign language, and then this circuitry developed, and then magically the circuitry gets co-opted in the vocal domain. Okay, and there's really no explanation really of how it is that you take these sophisticated manual abilities, which I'm not doubting that chimpanzees have. They certainly have gestures, and they certainly may be important. But how do you get from that domain of motor activity to taking that sophistication and then applying it to the vocal domain? Okay, so I want to try to eliminate that magic. Okay, and what I'm going to do is I'm going to say, okay, well, there's just something fundamentally different about chimpanzees versus humans, and I listed just a few of those, and I'm going to mention one other important difference in a moment. I'm going to say, okay, well, what, in terms of vocal behavior, is there another primate species that maybe shows greater similarities? Okay, and I'm going to kind of go all the way over here to New World monkeys, and this is where the marmosets come in. And I'm going to propose an alternative to this gestural hypothesis that I think is simple, but judging from the reviews that I get, controversial. And the alternative hypothesis is this, is that first, if you just have a combination of prosociality and volubility, volubility simply means that you just vocalize a lot, that combination basically leads to vocal cooperation and entirely skips this gestural step that many people argue. Okay? So how do I test this hypothesis? Well, I test this hypothesis using a species that is prosocial and voluble, and that is where the marmosets come in, okay? So one question you might be asking, well, why are humans pro-social and why are marmosets pro-social? And the general argument in literature is, which I support, is that these two species are among the very, very few primate species that are cooperative breeders. Okay, so what do I mean by cooperative breeders? I mean that not only do both parents share in the care of infants, older siblings will take care of younger siblings, and even unrelated group members would help take care of young infants. Okay? And the hypothesis, the more general hypothesis, is that this type of cooperative breeding strategy just changes the way that your cognition works in a way that allows more pro-social behaviors to emerge. So paying attention to others, and I would argue to vocally cooperate with each other.
But in that definition, aren't the mice we saw yesterday also cooperative breeders?
So there was no evidence yesterday that, for example, old unrelated mice in a natural situation, unrelated mice would take care of.
But they do. The females are taking care of the young, the pups of other mothers.
Right, but that was an experimentally induced care, and I think that argument will actually become important towards the end. Okay. Remind me if I don't mention it, but. Okay, so what do I mean by that they're pro-social? I've been kind of vague about that. So this is just a schematic graph that basically has strength of pro-sociality on this axis, and then on this axis is just strength of relationship. So you can have closely bonded kin, closely bonded non-kin, fellow group members, anonymous group members, and then true strangers. And so this is the extent of pro-sociality on this axis, the extent on this axis of the strength on this axis, and you have these different species, okay? And you can see that humans are very, very pro-social. You take a species that's closely related to marmosets and other New World monkeys, and they're not so pro-social. You take chimps, they're really not pro-social at all, but you see these marmosets are quite close to what humans are in terms of who are they willing to help in relation to how closely bonded they are to that individual in terms of kinship, okay? And there's actually a paper that just came out by the same group led by Burkhardt. It came out I think a few weeks ago in Nature Communications, where they took a test that's similar to the test that I'm about to show you, and they tested all these multiple species, cooperative breeding species, non-cooperative breeding species, and they asked, well, which one of them seems most pro-social? And they end up with basically this result, with all these species using essentially the same behavior paradigm. What does that behavior paradigm look like? It looks something like this. So this is a food sharing paradigm. And again, I'm just illustrating this to you to emphasize the fact that marmosets are pro-social. So in this paradigm, basically, there's an apparatus, and on one side of the apparatus is a marmoset that can pull a tool. And when he pulls the tool, it actually delivers food to the other side of the apparatus where there's another marmoset. And by pulling the tool, the marmoset does not get any food reward or anything for himself. His decision to pull only delivers food to the individual next to him. Okay? So they do this, and they can measure, what's the proportion of pulling for this food when the individual is closely related to the marmoset, the individual is unrelated to marmoset, versus there's no one there. So maybe the marmoset, so one possibility is the marmoset just pulls the tool because they like pulling the tools and it doesn't really matter. So they control for that possibility in the measure. So if you look at the data, it looks something like this. So you have a graph of how often do they pull without anyone being there, and then how often do they pull with a partner. And you look, and if you take look at all the different relationships, and they pull significantly more with a partner there than without a partner there. Okay, so they're willing to pull, again, without getting anything for themselves. And then if you split off from the individuals that they pull for, those that are not related to them, you still get this really fascinating effect where that individual is not related to them, but they're going to deliver food for them anyway. And that's really a kind of a remarkable demonstration of prosociality. Yes?
They're not just doing it because they're bored and there's something to do, even with something else there.
So the question is, are they doing this just simply because they're bored? And I think these guys would argue they may in fact be doing it because they're bored, and that would be the without partner condition. And so that's why they contrast the with versus without partner condition. You seem skeptical. Why?
Because without a partner and with a partner, if you have a partner there, there's another object in the room to interact with. So it doesn't need to be a sharing thing, and that might explain why there's no relating.
Oh, so that's interesting. So I hadn't thought of it that way. So the suggestion is basically, if I'm correct, is that it's interesting if there's not a partner there, it's interesting and you might pull at some level. And if there is another thing there, just because arousal is greater now, that you're going to just be more active. But that's a good argument. I don't know if they address that argument. True, but I think you could still make if you really want to, you could still make an arousal level, because it's not that they pull significantly more without a partner there for all kinship levels, but I think they still pull more for closely related kin. You can say, okay, well that induces more arousal, but it's not like they're, if you've seen a marmoset, they're pretty hyperkinetic, and so the fact that they're even pausing at all to pull is kind of a miracle.
But in terms of the related, non-related, it might not be statistically significant, but it's sort of either it's more for non-related partners or it's the same, not less, right?
Relative to related partners?
Yeah, less is related partners, no? Right is non-related partners. And you seem to be saying...
No, this is all subjects. So the left-hand one is all subjects related or not related. They're just demonstrating that they'll pull more if somebody's there versus not. Right. And then I've just split off the graph that shows the unrelated individuals and that they pull for them. Mm-hmm. I'm shortchanging their paper, which is more comprehensive than I'm presenting in this one slide. Okay, so with this in mind, and with the knowledge that that marmosets basically talk a lot, we basically decided to explore their vocal exchanges. And so I'm going to play you something that's, at least to my ears, is quite irritating. I'm going to play it once, but it's basically two marmosets exchanging their calls. Okay, I hope the sound is on here. All right, can you turn the sound on, please?
Can you knock on the window there and tell our friend behind the window to turn up the volume? Knock on the window, he might not get it.
You can say it loud and I can hear you.
No, it worked. It worked. They just have to turn the sound on because it worked when I.
Okay, should I sound?
There we go. Okay, so that's one marmoset producing what's known as a phi call. That's another marmoset producing the response. And then the first marmoset will respond again. Okay?
This is what it sounds like in the lab.
Yeah, and what the beauty of it is, once you get past the irritation, is that they will do this spontaneously, and they will do it frequently, which gives us a great model system to explore. And while these seem while they sound annoying and they seem structurally very simple, this is a spectrogram that's basically breaking the frequency components down over time, there's actually information embedded in it. So you can, the marmosets can extract information about, gender, individual identity, group member, age, and so on from these signals. They have individually distinctive characteristics into them. But we haven't exploited that aspect yet. What we want to understand is, are they using, when they do this exchange, how cooperative is this exchange? So I'm going to describe two behaviors that I think support the claim that their communication is cooperative. Okay? The first behavior is if it were humans and if I were trying to exchange or have a conversation with someone that was standing here versus having a conversation with someone that was standing 100 feet away from me, I would adjust my amplitude of my speech to facilitate the distance between me and this person that I'm wanting to talk to. Okay? Most animals, when they're alternating vocalizations, they're doing so in a very competitive context. Okay? So for example, frogs will alternate their vocalizations with other male frogs because they know if they're overlapping, they know, I'm anthropomorphizing a little bit, the system knows that if you're overlapping the call, then there's no way that the female is going to be attracted to any one of them because the female can't localize either one of them. Okay? And when the frogs get closer together, they try to outdo each other. They try to be louder than one another. Okay? So it's the exact opposite of a cooperative communication system where as distance increases, you try to say something louder to facilitate getting the message across. And that has only been demonstrated in humans. Okay? So we want to know, if marmosets are pro-social like humans, if they're voluble like humans, do they demonstrate this distance effect as well? So we basically ran an experiment where we simulated distance from a conspecific, a member of their own species. We simulated distance simply by playing back soft calls of low intensity, low amplitude, versus loud calls. Okay? Certainly, there are other cues to distance, but sound level is the primary cue to distance. Okay? So the setup looks something like this. It's quite simple. We have a room where we have a marmoset in one corner, a curtain, and then there's a speaker that's going to play back these sounds. The sounds are going to be, if it's a nearby sound, it's going to be 50 to 60 dB. So we're playing back these feed calls. So we're simulating the presence of another marmoset. And if it's a far away marmoset, the call is going to be roughly 20 to 35 dBs. And that equates to about being 5 meters away from a simulated conspecific versus 60 meters. And this is what the power spectra of those two far away and nearby calls look like. And of course, they look somewhat different because the high frequency component is missing, and that's simply a physics, basically. The sound attenuates. The attenuation of sound is greater for higher frequencies than lower frequencies.
That setup implies that the marmoset would produce vocalizations at a constant intensity. Otherwise, you cannot infer distance from intensity. Is that true?
I'm not sure I follow your statement.
No, look, in this case, you just say, look, the attenuation of the sound intensity should be interpreted by your subject as an increase in distance.
That's right.
Right? But that's only true when under normal conditions, these marmosets produce vocalizations at a constant intensity. Otherwise, intensity doesn't reflect distance.
Well, I think on average, it's a little bit what we're testing, but it's a little bit also an assumption. I think if I show you the data, then you will see that there's variation in their amplitude. And part of that variation may be explained by, under normal conditions, they have different distances from their conspecifics. I should have mentioned this earlier, that this particular call that I'm focusing on, and I'll be focusing on for much of the talk, it's called a feed call, and it's a contact call. So this is a call that they produce when they're isolated visually from other marmosets. So they produce this call, and it elicits a call from a conspecific, and then they've made auditory contact. And we can talk about the significance of that later. So this is the experimental setup. What is the kind of exemplar level result looks like? So this spectrogram with waveforms captures the entirety of what the result is. So here's the playback. This is the nearby playback. So this is the high amplitude call. So we do the playback, and then after a delay, the subject marmoset produces a response. Okay, so this is the response, long delay response of a nearby to a nearby marmoset. And then we play the far away call, which has a reduced amplitude. We see the response of the marmoset is not only quicker, but it's actually louder than the response to the far away call. Okay, so this is showing that instead of using a competitive mechanisms where, oh, I hear somebody nearby, and therefore I'm going to try to outdo that individual by giving an even louder call. They do what we do, which is they adjust their vocal amplitude in a cooperative fashion. And we can look at the population data, and it looks like this. So you have amplitude on this axis, you have the nearby condition, far away condition, and you can see for the far away condition, they produce louder calls. And it turns out, although it wasn't part of our original prediction, they're actually quicker to produce those calls in the far away condition than in the nearby condition. And so if you look at the human literature, so this is a very simple experiment with a simple outcome, but I think it reveals something important. Not only can marmoset monkeys flexibly adjust their vocalizations, but they seem to be doing so in a cooperative manner. Now, when people write about this phenomenon in humans, they invoke all sorts of special cognitive mechanisms, like, oh, well, if the person's far away, then I have in mind his mind that he's far away and that I should adjust my vocalizations because I can put myself in his shoes and can't hear me, so I'm going to adjust the vocals. So there's all sorts of kind of high-level cognitive phenomena that are usually invoked to explain why is it that we adjust our vocalization according to interspeaker distance. For the marmosets, I don't think, although some, particularly the individuals who do the kind of the pro-social food sharing experiments that I showed you before, they would like to argue some cognitive mechanism, and that may be the case for food sharing. But for this cooperative vocal control, I think it's possible to explain it much more simply using a homeostatic mechanism that's just based on arousal. Okay? And it goes something like this. So you have marmoset 1 here, and you have a node that produces a vocalization, and you have a drive where as the drive increases, the likelihood of producing a motor response in the form of vocalization is more likely to be elicited. And then you have input to audition, and the stronger the auditory input is into marmoset 1, the stronger is the inhibition onto this drive. Okay? So if you have two marmosets interacting together, and this marmoset is producing a high amplitude vocalization, it's going to activate the auditory system much more strongly, and that's just kind of a basic fact about auditory neurons. They tend to be intensity, monotonically tuned to intensity. The stronger auditory activation is going to lead to stronger inhibition on the drive, and therefore the marmoset is going to produce a lower amplitude vocalization at a slower rate. It's like the threshold is adjusted accordingly. And if you do the far away version, you have a lower amplitude, low intensity vocalization. The activation of auditory system is going to be less, and therefore the inhibition is going to be less, and therefore the drive is able to elicit a vocal response at a higher amplitude much more quickly. Okay, and now.
So why do you call this cooperative? Is that not overinterpreting the effect that you observe? Because so in some indeed, okay, feedback we agree, right? That's also what how you try to explain it. But in some sense, it basically means, okay, if this contact call, which is just says, hey, I'm here, right? It's not yet really, let's say, a communicative behavior, like let's do something together. If that contact call has a high intensity, you just answer with a low intensity and vice versa, right? So why would you interpret it in terms of cooperation already, that is dependent on also high-level representations and so on?
Okay, so that's a great question, and it's what I usually get in trouble with, which is how do I want to define cooperation? Right. Okay, and I think there's many ways to define it. So one way to define it, when people talk about cooperative breeding, they're defining it kind of in evolutionary terms, where cooperative means there's a fitness advantage to the individuals involved. Okay, and that's one level of cooperation. And I'm going to argue, and I'm arguing that basically that fitness advantage led to prosociality, led to vocal cooperation, with cooperation meaning something different, not a fitness advantage, simply a system that facilitates the exchange of signals. And so when I say here that this is cooperative vocal control, it's really having in mind that it's facilitating the exchange of signals in a way that's distinct from what animals typically do, which is try to interfere with each other's signal, try to outdo each other's signal. So I'm using two definitions of cooperation, and I apologize that it's confusing, but it's one of those things that I'm kind of working out as I go along with this the story that's developing in my lab.
Are you going to talk about variability in the data, or?
How do you mean variability detection?
I'm thinking, are there differences? If you can tell from the vocalization whether it's female or male, depending on whether you're male or female, does that affect your behavior? Or, you know, if there's one's the sound of a dominant male or something, and you're a submissive male, then do you behave differently? These kinds of things, because those kinds of differences would suggest that this simple sort of reflex loop is not accurate. And the other experiment maybe you've done already is also to say, okay, well, let's let the marmoset see the other marmoset, and let's test if I've got my guy nearby and he's shouting at me rather than being quiet. You know, I think, Paul's shouting at me, I'm going to shout back at him.
What are you talking about, Tony?
Yeah, so that's a great question. And, you know, students, you should know that when a speaker says that's a great question, it's because he has something to say. Okay, so that's a great question. So we've done this. So in this experiment, it seems like the features that change according to, there's many things I can say about this. First, in this playback experiment, the set of calls that we used were diverse. So they were coming from, the playbacks were coming from male or female, coming from pair bonded, unpair bonded, and it had no influence on this pattern of result. The second thing is the adjustments that a marmoset will make to its calls as a function of distance seem to be limited to this latency shift and amplitude. So if we look at other features, such as the number of syllables in the call, or the overall duration of the call, or the fundamental frequency of the call, they had no systematic relationship to whether they heard a nearby or far away conspecific. Now, to your question about have I done any experiments where they can where they seem to differentiate the potential information, individual variation information in their calls, here's one experiment that we did. So we had, instead of having, this was with 3 live marmosets in a corner of each in a corner of the room, and we just asked the following question: if you have this triad, who do the marmosets talk to? So you could have a male marmoset, it's pair bonded female, or a stranger female marmoset. And in that case, the male marmoset tended to exchange calls with the stranger female, as you might predict for a male. If you do the same experiment, if you have a female marmoset and her pair bonded male in one corner, and then the stranger male in another corner, she primarily converses with her pair bonded male. And what that experiment doesn't get at all the variables, but it tells us the marmosets can detect and use the individual variation information in their acoustic components of their signals.
Doesn't that then speak against this model?
How's that?
Because there's two
that's
female
right. Okay,
marmosets in the room, and you're
so this model.
behavior.
So this model is only modeled to demonstrate the possibility that you could have a simple non-cognitive mechanism to explain this form of quiet. It's not a model for the entire array of vocal behaviors that they could be exhibiting using this fee-call exchange.
So this is a straw man model that you're going to destroy?
I don't do that with my own models, but I have a more elaborate model that I'll get to in a few slides. Okay, but right now I want to say, okay, this is the setup. This is the kind of This is the kind of vocal control that they have, and now I want to get at, well, do they exhibit patterns of vocal turn taking that share some similarity to the patterns of vocal turn taking that we have? Okay. This experiment is in some sense more simpler, but has more complicated analyses. And in this experiment, we're just putting 2 marmosets in a corner, and then we're just letting them talk to each other. Okay? And so we're going to take these signals. They're in there for 30 to 40 minutes. We get these kind of signals, so sessions on the y-axis, time on the x-axis, and each hash mark represents a fee call from one individual. Red's one individual, blue's another individual. And then we're just kind of look at the natural statistics of these patterns. And then we compare these natural statistics to a permutation test where we mix the individuals across different sessions to get kind of a baseline control. And here's what we find. The first thing we find is that in this data set, which is thousands of calls between adults, they never interrupt each other. Okay, so the data is that green line. The probability of overlap or interruptions is along the x-axis, and then the permutation test data is in the gray. Excuse me. So they don't interrupt each other. And then you can ask, well, what's the temporal parameter of this? And you can see that they have this temporal rule, like roughly between 3 and 5 seconds is how long they wait before responding to another marmoset's call. Okay? So at a gross level, it's very similar to polite human conversations, but the time scales are very, very, very different, much, much longer. And we could talk about why it may be longer towards the end if you wish. Okay? So one of the ways you could characterize, sorry, there's a question there? The time scale of this is in seconds.
The one on the left. No, just like to better understand this. Like you have this time in the bottom.
Well, this is a schematic. So it's a schematic,
okay.
but you can think of it in terms of the seconds. So one of the ways you can characterize human interactions, including human conversational interactions, is you can characterize them as coupled oscillator mechanisms. And if things are coupled oscillator mechanisms, they exhibit kind of 2 characteristic features. One is entrainment. So the 2 speakers, for example, will be in antiphase with each other. So one speaker says something, then another speaker says something, then the other speaker, then the first speaker says something again, and they keep going back and forth in antiphase. So they're entrained with each other. And the other feature is they phase lock to each other, and the other feature is that they entrain to each other. So phase locking is when they kind of alternate back and forth, and then entrainment is when one speaker starts speaking slightly faster, then you will start speaking faster. If someone slows down, then you slow down. I'm sure a lot of you have had this experience personally. If you're talking to someone who's fast, you start talking faster as well. Maybe not as fast. Maybe not as fast. So we wanted to know whether these marmoset exchanges could be characterized in the same way that people have characterized human speech exchanges. And the way we did that is in the following, which is just to create probability distributions of the following. So the first question we're going to look at is, do they show the phase locking characteristics of coupled oscillators? So we use as a reference the vocalization of marmoset 1, and then we ask, well, when do these subsequent vocalizations in this session of 30 to 40 minutes, when are the subsequent vocalizations by marmoset 2 occurring? And then we take the second vocalization of marmoset 1 and do the same thing. And if there's phase coupling, then you'll see a distribution that has bumps and regular bumps in it. And if there's no coupling at all, then the distribution would be flat. Okay? So we do this experiment, and we can see that they seem to be phase locked to each other. Okay? So that's a bit of the easy part. The harder part is, are they going to show entrainment? If one marmoset starts speeding up, will the other marmoset start speeding up its turn taking? If they slow down, will the other marmoset slow down? And we can look at this, and we've confirmed this with playback experiments. We can look at this in the following way. We say, okay, well, marmoset 1, if it's just spontaneously producing its call, has this interval between its call. Okay, so this is T0. And then we ask, well, if this interval is interrupted by a second marmoset, is there a relationship between the timing of the marmoset 2's response and the subsequent call of the marmoset 1? Okay? So it's a phase analysis, and here's the hypothetical outcomes. Okay? If there's no relationship between how fast marmoset 1 responds and the response of the other marmoset, then basically there's no correlation and you see a flat line. Okay? If marmoset 1 speeds up and marmoset 2 therefore slows down, then you'll see a negative correlation. If they do what humans do, then when marmoset 1 speeds up, marmoset 2 speeds up. When marmoset 1 slows down, marmoset 2 will slow down. The result is that they're much like the humans. So the data are showing this positive slope here, where it's demonstrating that the marmosets, in essence, entrain to each other. And the positive slope stops here because after about 12 seconds, for the timing of the way marmosets produce a vocalization, if after 12 seconds you produce a vocalization, it's so long that it's unclear whether was it a response to the previous vocalization or are you just spontaneously producing a vocalization for some other reason. Okay, and that's where the correlation just goes away.
But the range of phase coupling is pretty broad, right?
That's right. And what's interesting, in at least right now, my understanding of it's too complicated to go into, is we've done a playback experiment. So this is just natural exchanges. We've done a playback experiment where we simulate the other marmoset, and we can adjust, we randomize the timing of that response. And we get the same result, except this curve continues to go all the way out to 25 seconds. So there's something else that they can pick up, or there's some limitation on a real marmoset that cuts the curve off at that.
If coupled oscillators, you would expect that the phrase length would also become correlated. But if you entrain two oscillators, right, it does mean their phase starts to become comparable in a similar range, which for the vocalization might imply that your phrase starts to get
Yeah, I see what you're saying.
the phrase of the conspecific. Do you see that, or you don't know?
We see, so we haven't looked at exactly that, and that may account because they will, they don't, if a marmoset won't always consistently produce a 2-syllable feed call or a 3-syllable feed call, so there's variation. And that's a good question because we haven't looked at, maybe the natural variation of this adjusting the speed is simply a function of the length. We haven't looked at that yet. That's a cool idea.
So if you had human data on there, would humans show as much variability as the marmosets?
Leah wants to know where she would be on this graph.
I can say on the right is the text messaging for it drops off quick.
The short answer is I don't know, because nobody has actually displayed it like we've displayed it here. They've shown entrainment, but it's only kind of just catching on in the speech literature. So we haven't actually looked at it, speech specifically, but there's certainly entrainment in terms of you speed up, other person will speed up, but the variation I don't know. So these behaviors are particularly interesting, and this is dovetails nicely with what Gary was presenting. You're getting this without a big brain. So one of the arguments from people who argue for the gestural theory is that they argue that well part of this cooperation must like all fancy cognitive skills, people argue, it must come from a big brain. And the same is true when people think about cooperation. But as you can see in terms of relative size, the marmoset brain is actually quite small. And what that tells us is there must be something really that's about the circuitry as opposed to simply the number of neurons. And we're very much interested in pursuing what is the neurophysiological basis of this interactions, and we're currently collecting data, but we haven't analyzed it. But it's a huge undertaking. And it's a huge undertaking because not only getting cooperation across individuals, so we're talking about, you know, how do two individuals basically synchronize each other, but just the act of producing a vocalization within an individual requires the coordination of respiration, tension of the vocal folds, and the opening and closing of the mouth. And that has to be coordinated before you can have coordination across individuals. And the number of structures involved in that process are numerous. And this is just some of those structures labeled in coded fashion here, but I'll try to. So for example, drive, you know, the arousal mechanisms that's going to generate a vocalization initially could be influenced by things like the amygdala, the insula, the anterior cingulate, which I'll mention again. Motor output, so you drive and then you activate a motor structure that's going to elicit a vocalization. So, you know, one prominent area is the periaqueductal gray area. And then you can have a corollary discharge mechanism, so, you know, an efferent copy that's going from the motor structures to the auditory system, and then you have another kind of copy of the signal that's going through the air. So you get an efferent copy through a direct connection, and then you have the signal that's going through the air through your own ear and then back into your auditory system. So that's another pathway that becomes important. And then the auditory system then kind of interacts with the drive system. And these connections that I've put here really refer to what we know about the anatomy and physiology of the anterior cingulate and the auditory system, with some vagueness with regard to the motor structure. But these connections are based on what we know about the neuroanatomy of those structures, the limited stuff that we know about the neuroanatomy of those structures. Okay? The reason I'm presenting this is because we wanted to generate a computational model that could give us more specific hypotheses than just, oh, I'm just going to put my electrode in, let's say, the anterior cingulate and I'll see what happens. Okay? So we built this model. The model is, you know, it's a circuit level model, but its activity is actually based on an Ishkovich model, which is the single model, which is the model of a single integrate and fire neuron. Okay? But what we're doing is we're treating the fire or the action potential as vocal output, and these inhibitory and excitatory things as currents in the circuit. Okay? So the output of the model would look something like this, where you have arbitrary activity on this axis, and you have the drive that's fluctuating at some level, and then it reaches a threshold, which is going to activate the motor structure, which is the vocalizations are indicated in red action potentials here. And once that motor is activated, then it's going to activate the auditory system, and the auditory system is going to get activated, and it's going to clamp down on the drive and prevent a vocalization. And there's some timing delay mechanism related to that. Okay, that's the general feature of the model. And then what we did is we take this model and we adjust the weights so that it fits the statistical patterning of marmosets by themselves producing vocalizations. Okay, so we did what everyone does with the model. You generate the model and then you fit it to some data. Okay, in this case, the data we fit it to are marmosets by themselves. They're not interacting with any other marmoset. They're just producing spontaneous vocalizations at some particular rate. We adjust the model to capture that distribution, and you can see here that, you know, this is the probability of producing a phi call. This is time on this axis. So the peak is around 10 seconds. And this is the number of syllables in those phi calls. This is the distribution of those syllables as a function of frequency. And then you can see that we were able to tune the model quite well to capture those statistics. Okay? That's actually, at least in my mind, as a person who's not really a modeler, that's not particularly interesting. Maybe somebody finds that interesting. I think my postdoc thinks that's interesting. What I think is interesting is, what if I take that model, what if I take this model, I tune it using those spontaneous vocalizations of an individual by itself. But what if I just take this model and then just pair it with another version of the identical model, okay, without doing any kind of tweaking whatsoever. What if I do that? Okay, so it looks something like this, and the way this model interacts is that when this side is producing motor output, it's activating the auditory system of the other individual, which can influence the timing of the drive mechanism of that other individual. Okay? And what we find, I think, is absolutely remarkable, although this is where, the expertise in people in my lab far exceeds mine, especially in this domain. What I find remarkable about this model is without any further tweaking, we basically capture all the turn-taking data that I've shown you so far. So if we do this model, the probability of overlapping is basically 0. The timing of the vocal exchanges is essentially the same. So the model's in blue, the data are in green, and you can see the timing is roughly between 3 to 5 seconds. If we look at the phase locking, you see that there's similar phase locking with a similar timing component in the model versus the real data. And the model will also produce, although not exactly identically, the phase response curve. Okay, I think that's actually remarkable, and this gives us, generates ideas about, okay, well, what is the output of this model in terms of the simulated neural activity of the drive structure versus the auditory activity, and then we could say, okay, well, now we can record in those structures and ask, well, does the model correctly predict the patterns of interactions between, let's say, the anterior cingulate and the auditory cortex? The other thing the model does is it allows us to do the various kind of lesion experiments, but computationally. And it turns out, for a variety of reasons, this connection, the efferent copy between the motor structure and the auditory structure that's illustrated in the normal model above, seems to be absolutely critical for capturing this vocal turn-taking behavior. Okay, so if I take this connection, this efferent copy here, and this copy from auditory to the drive, if I take that circuit out and pair the two models together, or even if I have a normal model on this side and this impaired model on this side, what happens to the turn-taking behavior? It basically all falls apart. Okay, now you suddenly have a bunch of interruptions. The timing is now completely off. And although there's phase locking, the pattern of phase locking doesn't account for what's normally seen in the natural data. Okay?
But that might be parameter dependent, right? Because as long as you have some sort of, if you have two coupled oscillators, you must be able to replicate your data. As long as you have the freedom to fiddle with the parameters and the transduction delays that you have running within your model, right? So have you looked at that whole complete parameter space?
So we have not looked at the complete parameter space. What we're trying to emphasize is if we tune the model in that first step where they're not interacting, and it captures beautifully a whole set of distributions about the properties of the call, not just one behavior, for instance, but the timing, the number of syllables, so on and so forth. And then we don't do any tweaking and put the two models together, and we get the result that recapitulates the data. We think that's pretty fascinating. And I think what you're asking is, if I have just this model, is there any, without the efferent copy, is there any way that I could tweak the parameter space and still get the original result?
Well, I'm more saying you might be overfitting your data, and with a more minimal model, you could have accounted for it as well. And then the lesion would not have been relevant anymore. It's a typical problem you face with any model.
That's right. So the short answer is we haven't fully explored the parameter space.
Well, I have a different question, but Paul is saying in some sense you haven't fully explored model space. So aside from not fully exploring, is this on? Aside from not fully exploring the parameters within your model, he's saying there might be simpler models that can capture it, I think.
Yeah, exactly.
My question is different, which is, do you have any reason to think that this gets the phylogeny right? So is it the case that the species that don't do the turn-taking, maybe we don't really have that data, but we can guess at it, are the ones that don't do the self-monitoring? Is there any independent data about who might do the self-monitoring or why?
No, so this is another a good point that reviewers point out to me frequently, and it's a good one in the sense that I'm making many claims about cooperative breeding, about cooperation in another sense with vocal turn taking, and now I'm saying something about self-monitoring. And the strength of the claim is only as good as the comparative data, which is what I think you're suggesting it. And we don't have the comparative data.
I mean, you could turn that around slightly more positive and say another source of evidence that you could use to constrain the models would be some comparative data. So in some sense, what Paul's saying is it's under constraint. It's not that hard to get these kinds of phenomena out of some kind of model. And there are different possible sources.
Yeah, so I think
of information that might
your question made me understand the point a little better, so I can maybe answer it in a different way. In other species where there's alternating calls, without calling them cooperative, let's say frogs and crickets, they basically use the exact same conceptual mechanism, where there's some aspect of self-monitoring, or they wouldn't call it self-monitoring, they call it efferent copy, that allows the animal to distinguish its own calls from that of another, and that adjusts the timing dynamically and somewhat simply. Okay, so for the way we develop this model, knowing that there are probably many ways to generate the same phenomenon that have again been shown in frogs and crickets. What if we assemble the model based on what we know about the neuroanatomy, and how would we get that phenomena with what we know about primate neuroanatomy and related to vocalizations and hearing? And so,
right. What I'm saying is you'd have a stronger case if we can say this is why we know that, say, the frog neuroanatomy is different, and these are the predictions that we make if you don't have this pathway or something of that sort. That would be the kind of thing that would make it feel like it's not just that these are relatively easy data to model, but that they're making some predictions about the distribution across.
I take your point. It's a good point. We don't have that data. You know what I need is a neuroanatomist. Not only a neuroanatomist, a good neuroanatomist. Okay, so let me just, when I give this talk in front of ethologists, here are some of their objections. Okay, so they say, well, but frogs and crickets alternate their calls too, so what's the big deal? And my answer to that is, well, marmosets do more than just calling and respond in mating in a territorial defense context. They seem to be cooperatively taking turns with other individuals. Okay, so it's not a competitive thing, which is the case for those other species. And then say, well, what about duetting and songbirds and gibbons, which are another type of primate, and they exchange with their mates. They exchange very exquisite orchestration of their vocalizations. It's really, really beautiful. And it's almost impossible to tell that there's two individuals producing these sequences of calls. So people say, well, isn't this just like the call coordination of those species? And the answer to that is, it's like that, but for humans and marmosets, they can have this call coordination with any other individual. They don't have to be pair bonded to that individual. They don't have to be of the opposite sex. They're willing to have this coordinated vocal exchange with any other individual. And I would argue that comes from the fact that the underlying basis for their behavior is prosociality. Okay? And again, to Gary's point, to really get at that, what you need to do is, well, what about a closely related species that doesn't have cooperative breeding that may exchange vocalization? Do they exchange it in this pattern or in a different pattern, so on and so forth?
The last couple of slides make me feel like maybe you'll find coupled oscillators pretty broadly, and it's really about what serves as the input to the coupled oscillators.
I think that's right.
Or
I
the
totally
domain
agree
in
with
which
that.
you use them.
Yeah, and I said I have a review paper out.
I mean, certainly there are other parts.
Yeah, I have in ticks, I just published something that says exactly that. You could have the same phenomena, and it could be instantiated in many different ways depending on the species and the input.
And what you're looking for is like a pathway, connection to a social pathway or something like that, if you want to understand.
No, absolutely. And that's where we're going, provided we can find this good neuroanatomist.
And good meaning he agrees or she agrees with you.
No, not necessarily. I change my mind a lot.
It means he or she is willing to go along with the changes in your views.
That's right.
So where you seem to be going with this is that you're showing nice data that suggests that calling in marmosets supports their cooperative behavior and their pro-sociality. But it doesn't have defining features of language, that there's a message that you're communicating and you care about whether the other one is getting that message. I like the models. They're really nice models and elegant. But if I was wanting to argue that marmoset language was, like early hominid communication in some way, I would want to say, well, marmosets break this oscillator model, because like we said, other species have oscillators. It's not a big deal. But I would want to know, do marmosets do things which overrule the oscillator model in such a way that, if there's some particular thing about what I want to communicate to you, that I don't act in a way that is predicted by the oscillator. There's some added feature of the data which is not explained by this model. And then I would get excited, because I would then think, maybe the marmoset has some theory of mind. Maybe the marmoset does know that you don't know something that I know, and I want to get it across to you. And then that's language.
Yeah, I agree with everything you're saying, except what you think my goals are, which is I don't think there's anything in non-human primate communication systems that's anything like language. What I'm trying to establish is what could be the base level foundation to develop a language. And I believe that it has to start with some sort of cooperative form of communication. So for example, macaques don't do anything like this. There's no evidence that chimpanzees do anything like this. So we have to think of a different way where you could get that cooperative communication, and that's what I'm trying to demonstrate here. And you're right that when we have a conversation, we can be phase locked to each other, but one person can say something that makes you pause, right? And then you break the thing. The marmosets don't have any sophistication like that. What makes them distinct from just frogs and crickets may not be the fact that they're acting as coupled also, but that they also communicate in this cooperative way by adjusting their vocal amplitude. That's kind of one of the motivations for doing that experiment.
Quick question, but in the context of like validating this, can you perturb that behavior? Can you perturb the, parameters of the system, just modeling that phase coupled behavior with, say, a danger signal, or, putting in a natural, something that's preying on him in the cage or something like that, or exciting him in some way, and then seeing that perturbed, and you could measure that?
So these last two questions made me think of a possible experiment that may be able to answer, that would be satisfactory experiment. And that is, let's say I do the playback, because you can generate these patterns just using playbacks, right? It's an interactive playback. The computer detects when they call, and we can time when the computer plays back the call. What if in that experiment I switched who the speaker was coming from the playback, who the individual who calls were using coming from the playback? And the question is, would they change the timing would be the same, but would they first of all detect there's somebody else calling, and would that disrupt their locking? I think that would get both your questions, and it might be a cool experiment and an easy one to do, actually.
Something along the lines of testing the model, as well as you have one real marmoset, and then you have one synthesized based on your model, and see if the real marmoset could have a conversation or a communication in an interactive way, in a manner it normally would do with the natural marmoset.
Yeah, so it's funny that we're doing that exact experiment, because the model leaves a very strict predictions. If we simulate this, if we drive the interactive playback engine with our model, the question is, do you get the same natural statistics? But then we could do one additional step. What happens if we lesion the model? And the model has because in our model, we can have one intact version of the model and one lesion version, and that outputs a different result. And we can then ask the real marmosets if that output, if you lesion the engine, the model that's driving the engine, if you lesion that one connection, are you changing the behaviors of the marmoset? And that would lend a lot of credence to the model, I think.
It would, but with the caution that you have to care about the parametric. It would, but with the caution that Paul raised before about parametric space, I mean, you really have to make sure that the predictions are robust against variation in the model, basically, before that becomes convincing.
Sure.
I don't know if this is the right time to bring this up, but we can postpone it if you think it derails the whole story. But
Depends on if I can answer it or not.
exactly. So my question is a little bit about whether we're really looking at a specialized mechanism for coordination, or whether we look at the co-opting very generic features of nervous systems for now this specific function. Like for instance, take oscillators. If it's your spinal cord, you will find them in your thalamus, in your cerebellum. They're all over the place. Buzaki has a beautiful story on the whole brain, being organized around
Sure.
oscillators. You look at entrainment. There's a very common feature in brains to find entrainment. You can use binaural beats to entrain the thalamocortical system. So that just means you have two sinusoids, slightly offset in phase. You will experience hearing clicks, and the frequency of those clicks you can recover in the EEG. You can think about steady-state visually evoked potentials used in brain-computer interfaces, where you have flickering lights. You recover that flicker frequency in primary visual cortex and use it to drive a computer, right? So there are a lot of examples that oscillations and entrainment are a very generic feature, right? Or you can go back to, let's say, insects with our cast and search strategy to follow chemical gradients, right?
Yeah, no, I agree 100%.
So the point is it really such a specialized circuit or system feature we're looking at as you try to make it here, where you say, well, I have to hear this specific call, then it has to impact some sort of arousal system in a very specific way, and it has to trigger specifically another call. Maybe what we're looking at is just the use of a very generic property of brains that are applied in many different domains of behavior and perception. But now in this case, for, let's say, communicative or cooperation function.
But I think so I have a whole nother talk that's just about theta oscillations and theta structure and signals. I'm on board with there's many different oscillations and many different types of rhythmic activities, but all of those aren't being generated by the identical mechanisms specifically. Although there's excitation, there's inhibition, certainly. Okay, and you can generate these rhythmic behaviors, and they come in different forms. With regard to the specificity, if I take a marmoset and I do a playback experience with a different call, they don't do this behavior. So there is some specificity in this system.
But that's not due to the neural mechanism. That's due to the content of that call, the pitch and timbre structure of that call.
Well, how is that pitch and timbre structure extracted? It's by the nervous system.
No, but the process that allows this extraction is not qualitatively different from a structure that might generate action, right? It's just that the input filters into that structure are different. And that gives you the ability to extract pitch and timbre. So now suddenly it becomes a call-oriented circuit, subcircuit.
Yeah, I mean, I don't think I would disagree with you. I don't see any reason to disagree. There's
that's fine.
There's multiple oscillators in the brain. There's no doubt about that.
Okay.
This is just one. I'm not saying it's a special form of an oscillator. I'm just describing one way that really captures this interactive behavior across two individual marmosets, and those individual marmosets are oscillating by themselves as well.
That's fine.
In my talk, I made a bet, I guess. I had said that language would be something old and something new. We can even put the something new aside for the moment and say that a lot of what goes into language would be old components, but they won't necessarily be configured in the same way in our closest cousin. So it might be that all of the elements of language are things we see elsewhere used in different contexts. So you might see oscillators, you might see categorical perception, you might see sequencing in different places, but we somehow put them together uniquely. And so, even if oscillators per se are not exciting, they're not unique, nailing what those pieces are in language is really important. And finding animal homologs where those pieces are used in particular ways can be informative. So it doesn't have to be that the right answer to language is there are 4,000 new mechanisms. Probably a lot of them are old and just used in new ways.
That's right, and I think that kind of speaks to a question you had earlier. You could have this. I think that was something that wasn't addressed in your talk, but you're probably sympathetic to, is that you can indeed have the same circuit that could just be modulated slightly differently. The connectivity diagram is identical, but you modulate it differently and you get a totally different behavior. And that's been, again, by Eve Marder and people like Paul Katz, studies like sea slugs, to show you can get remarkable differences in output with the exact same connectivity. And that's totally missing from all the brain initiative hype.
Just a small question. These marmosets have been born and raised in the laboratory.
That's right.
So all your conclusions on this?
Well, so it's impossible to do the experiments I'm doing in the wild, but they certainly exchange vocalizations in the wild. And I have a colleague named Corey Miller at UC San Diego who's outfitting his marmosets in the wild in Brazil with little collars that record with the microphone, and he'll be able to say definitively whether it's...
But so it's not sure whether this turn-taking, for instance, also is true in the wild, or if there is any evolution in the language in the laboratory, for instance, where the only contact is between the marmosets and the caretakers, for instance.
So there's many ways to answer it. So first, we know from experiments, from field experiments in the wild, that the marmosets exchange vocalizations. What we don't know, because no one's done the analysis, is do they have this coupled oscillator mechanism and all the details that I've shown you before. The way they're raised I have a breeding colony in my laboratory. They're raised in family groups. They're housed in family groups, and then different families are housed in different cages. And their proximity, the fact that they're in family groups and that their proximity to other groups is similar to what's seen in the wild. But there's a zillion other things that we, of course, can't. I have no reason to think that what we're showing here would be different in the wild.
It's a really interesting species in which to look at this vocal communication. And I just think maybe there's a lot more to get out of this system. You'll know Robin Dunbar's theory that language arises out of the need to maintain social relationships with a large number of other people. So he suggested that if we use grooming the way that chimpanzees do to maintain their social hierarchies, then we would be spending all our days grooming each other. So conveniently, or in order to get around that, we evolve language so that we can maintain our relationships in a much more efficient way. So it would be interesting to know with marmosets, do they have large troop size? Do they have interesting social hierarchies within the troop? Do they, does their sociality extend to having complex relationships within the troop? And therefore, are they going to be using these calls to maintain their troop relationships in the way that Dunbar suggested? I mean, do you think that's something that's
so that's a great question. I'm very sympathetic. Very sympathetic to that specific idea. I'm very sympathetic to that aspect of Robin Dunbar's idea, and I relate it to probably this is going to elicit a strong reaction. I think that the foundation of this cooperative communication is basically arousal reduction. So that's what grooming does basically. It releases stress, it reduces stress. And while we don't have the answer to your question, we don't have it in marmosets because the nature of the way we house the marmoset precludes doing something like that. The experiments that we did with lemurs directly addresses Dunbar's hypothesis, actually for the first time. The hypothesis is there any relationship between those individuals, or part of his hypothesis, is there any relationship between those individuals who you groom and those individuals who you interact with vocally? And it turns out in lemurs, it's almost a direct overlap. You groom these 10 individuals, and then you either monitor their natural vocal exchanges when they're out of sight with each other, and they're exchanging vocalizations only with those individuals. And you could do a playback experiment, and you take one of the lemurs out, and you do a playback of that calls. Only the individuals that he grooms or she grooms will call back. And you do a social network analysis. The networks are highly correlated with who do they vocalize with. That network is highly correlated with who they groom, and not correlated with other variables like play or relationship or kinship and things like that. So we're very sympathetic to that aspect of Dunbar. I keep saying that aspect because I'm not sure I believe the whole social brain increases with increasing social complexity. But yeah, Dorn.
You might want to comment on the fact that it's just a comment about the breeding system of marmosets, that you might want to comment that these individuals are maintaining close spatial coordination in their travel through a rather small territory during the day, and these calls are one way to maintain coordination when they're out of sight of one another, for example, and so forth. So it's not, I think Dunbar has an interesting idea, but he sometimes overlooks the problem of coordination, which is not just a matter of number of individuals.
Okay, so one thing you might be asking is, well, why do they bother turn taking at all? And one is this idea that they're reducing stress or arousal, or it could be rewarding. That's another aspect that we'd like to look into. And another aspect that's not exclusive is that they're just exchanging, you know, in order to exchange information in a noisy environment, you don't want to overlap your calls because that's interference. And you want to have enough gaps so that you can hear another person's call maybe repeatedly so you can extract the information about who the individual is, what gender they are, so on and so forth. Okay, so now I'm going to switch gears a bit. How many hours do I have left?
Three, but you got to hurry up.
Okay. How much longer do I have?
You only have like 5 more species.
10 more minutes.
No. We started 12:15, so, you know, you
Okay,
have
so
90 minutes.
I'm going to race through the most interesting part, which is how does this behavior develop in marmosets? And the reason why this is an interesting question is it turns out that when marmosets are born, they can't do this vocal turn taking. That made us investigate, okay, what are they doing when they're born? And the assumption for most people who are interested in the evolution of speech, evolution of language, is that they say, listen, non-human primates, they're not vocal learners. They're just born. They have the quote-unquote innate ability to produce calls that sound like the calls that they're produced when they're adults, and that those calls can't be changed with postnatal experience. Okay? So we wanted to first look at, when most people say that something is innate, they're basically saying it doesn't require experience, or there's some genetic blueprint, but basically it's another way of saying, I don't understand how it develops. Okay, so we wanted to investigate that further in marmosets. So just to give you an overview again, for the human literature, we know there's fetal crying that's highly correlated with postnatal crying, and that can lead to the first types of contingent responses with a caregiver, which may ultimately lead to learning how to, or scaffolding the ability to take turns. And so what I'm going to show you is the same, apparently the same sequence, but with marmosets. And we're going to start off by looking at ultrasound data and then tracking marmosets longitudinally, individual marmosets longitudinally, to watch their vocal behaviors emerge. Okay? So the idea is that we're getting at the origins of quote-unquote innate monkey calls. Okay? So this is a huge advantage of having a breeding colony of marmosets, is that we can watch them develop non-invasively with this ultrasound technology, and then when they're born, we can watch those same individuals as they grow up within their family groups and the larger social groups, and see what, if any, are the relationships between these different developmental stages. Okay? So just kind of an overview. I'll try to be quicker about this, but basically it's a standard ultrasound machine. But since marmosets are about this big, our probe is much, much smaller than what's used in a hospital on humans. And all our ultrasounds are focused on the face region right here. The gestation length of a marmoset is about 150 days. The face is actually visible, like developed enough that you can identify it as a face at around 94 days. And we just have, it's 5 days a week, 30 to 40 minutes. The mom is awake and restrained. She's habituated. She's used to being restrained while we do the ultrasound. She gets marshmallows during breaks, which looks like the cocaine of the marmoset world. And so we can do this, so the moms are awake and we can watch these fetal behaviors, and we do this almost every day until they're born. So we have more data than we know what to do with. I don't know if this is, so this is just to orient you. I'm going to show you one video of the type of things that we can see. So this is the head of the marmoset. You see the two eyes here, and the mouth is right here. And these kind of oral facial movements we can start detecting again around day 90. So here, hopefully, is an example of, you'll see a little type of chewing movement in this video. Okay, the mouth again is right here.
That's when they get the marshmallow.
That's right. Okay, so that's one example of the type of things that we can see with these videos. The first thing that we observed, which is kind of a general phenomenon in embryology and fetal behavior, is the embryo or the fetus basically starts making spontaneous movements, but they tend to be general kind of whole body movements that later get differentiated. So what we wanted to first track is when do oral facial movements become separate from more general body movements? And as a proxy for more general body movements, we tracked head movements. So I'm going to show you an example of a video at gestational age 102, and then the same individual, or at least the same pregnancy, at a later gestational age. And you'll see in the first video, there's going to be jerky movements when the mouth, jerky head movements when the mouth is also moving. And in the second video, you're going to get mouth movements without any head movements. So can everyone see that? Right, so the mouth is right there, and the head is kind of jerking back and forth, and the mouth is opening. And then a little later on, what you'll see, this is the mouth right here, the two eyes, and you'll see the mouth moving without the jerky head movements. Yeah, it's like a yawn, basically. Okay, so we can basically, using computer vision algorithms, we can basically track these movements, and you can see, so for this particular example, we can track the opening and closing of the mouth, and then the movement of the head relative to a landmark, and so the mouth is this line, and the head movement's on this trace, and we just want to track what is the development of these two behaviors, and when, if they do at all, when do they differentiate from each other, okay? So here's basically a raster plot of how many mouth movements in red, head movements in blue, how many movements do they make per minute over gestational age, and what you could, I'll show you more clearly, but basically what you can see is that there's a lot of head movements early on that kind of fade out, and then there's kind of a peak of mouth movements in this part of the raster plot, and you can quantify that here. So you have number of movements on this axis, gestational age on this axis, and you see this inverted U-shaped curve for mouth movements and this kind of linear drop for head movements. And this is consistent across the 4 pregnancies that we track, this overall pattern. And this U-shaped curve is quite common, and we're not sure, whether it's what's when cortical inhibition starts coming in, or whether there's constraints within the uterus that prevents the movements. That's an interesting question. But more importantly, the setup is over time. These are all the instances in purple are when you have this combination of head and mouth movements. And what we're showing is that those overlapping movements decline over time. And so the head movements and orofacial movements are differentiating from each other. Okay, and that sets up now our focus on what's going on with just the orofacial movements. Okay, we know from the human literature that the human fetus produces a variety of facial expressions that seem to have a corresponding facial expression postnatally. Okay, and we wanted to basically track now our different pregnancies in that same individual across postnatal days. So here's an individual that, if you look up top here, The two eyes are right there, and the mouth is right there. This marmoset fetus seems like it's producing a feed call. Okay, it's not, it looks like a cry, actually. And now the same marmoset on postnatal day 1. And then we can continue postnatal day 7, and then as an adult, although I'm cheating on this one, this isn't actually the adult version of this one, but they,
Don't tell us that. Don't break the illusion.
it's just it's very much what they would produce, okay? So that's kind of just exemplars of what we're seeing, which is basically the emergence of oral facial movements, which will be the oral facial movements that they will use to produce these vocalizations, okay? And now what we want to do is kind of track more quantitatively. That's just, those are exemplars, and it's always easy to find the exemplar, but harder to find the actual pattern. So what we did is we took a look at, on postnatal day, what are the different vocalizations that they produce. So they produce, for example, feed calls, and then I'll show you a little later versions of the feed calls and Twitter calls and so on and so forth. And we just looked at some of the statistics of those calls and see whether we can find matches to those statistics in the fetal videos, okay? So if you look at the statistics of those calls, they look something like this. So you look at duration or number of syllables, and you can see that the fee call is very distinct in terms of the number of syllables and the duration from the other calls. And if you look at the Twitter, if you combine the characteristics of duration and syllables together, then you can distinguish Twitters from the other calls as well. So this is the pattern of those acoustic features on postnatal day 1, and now we want to see now we want to use this information to classify the different oral facial movements that we see in the fetal videos. And what we find is, if we look at the percent of movements that are fee-like both in duration and syllables, we see that those percentages of fee-like movements start to emerge and then become most numerous towards the end of the gestational period, and the same is true for the Twitter. So this is just one way of showing, and we want to do a lot more sophisticated analyses to demonstrate that how it is that you get this, when the marmoset's born, it's producing these calls, what are the developmental processes, at least in terms of the oral facial movements, that lead up to that? And the more sophisticated analysis we're doing will be based on data like this, where we can track on infant P1 what is the temporal pattern of a fee call mouth movement or a Twitter mouth movement, and then find corresponding ones in the fetus and see how well you can predict one from the other. So more statistical matching there. Yeah.
Just state the obvious, that this really puts an interesting bound on these arguments about innateness. So you need this, presumably these are not learned. Maybe someone will want to argue this, and presumably there's some calibration that happens postnatally, but to have that level of motor control before postnatal experience, I think is impressive, and put some boundaries on what the computational toolkit needs to be.
That's right, but I think it all depends on what your definition of learning is. So the people have done experiments in rats, actually, where they can go in surgically and yoke the two legs together, for instance. And if you do that in utero and you do that for a particular time, there's the probability that those two legs will operate together increases, and they would argue that that's learning, basically.
I don't want to deny that there's any learning in the womb, but is there learning to sort of shape this particular behavior to take on the character that it does? Be surprised if there was.
I'm going to remain agnostic on that. I mean, there's a lot of experience in the womb. A lot of that experience is self-generated experience. There's a whole literature that suggests that spontaneous movements, and I'm sure you know this, spontaneous activity in general helps shape circuits, and that changes the spontaneous activity in a different way, which helps change the circuit. So especially with movements, like Mark Blumberg, for instance, talks a lot about twitches during sleep that help shape the somatosensory system. And there's work done by Bujaki's group, like Rustam Kazipov has done work both on rats and premature human infants. They haven't demonstrated definitively, but they provide a lot of suggestive evidence that these spontaneous movements help construct circuits, and therefore it's a form of experience-dependent learning that I would argue is not so different from the other types of experience that they may get postnatally. Okay, so what I want to kind of close with, I think, is the following, which is if you look now at postnatal day 1, so that's the kind of, what I showed you is the fetal story, and obviously it's developing, and so is this story, but it's a little more compelling in its message. When we start off, most people think that when a non-human primate is born, they're basically producing very clear versions of their adult calls, okay? And it turns out that in marmosets, at least very early in development, nobody had ever looked, and it turns out in marmosets, they don't produce very clear versions of their adult calls. So what they produce early on is a cry that looks spectrographically something like this, and then it transitions into kind of a half an adult call with a cry, which we call a phi cry, and then it can turn into, at least for this, I'm showing it only at one syllable level, then it can turn into a mature-looking phi response, okay? And you can measure various parameters of the cry and the phi cry and show that they're overlapping with the phi. And so here's just one example. So if you just look at the duration of that one syllable, on week 1, the twitter is kind of the control. So the twitter doesn't change, and it's a very short duration syllables. Then you have phi cries, phi, and cries. And you can see over time, these distributions start to overlap. So it's almost like the cry it seems like the cry is turning into the phi, and that's very similar to the way certain speech sounds are produced, and it's also very similar to what people have reported, people like Over Chernikowski have reported for development of song syllables and for zebrafinch. So what we basically measured is, what is the proportion of cries over time versus mature sounding phis over time? And you can see that there's kind of a switch. Very early on, infants will produce, this is an example from one infant, the infant will produce lots of cries, and then there's a transition where they just stop producing cries and start producing phis. Okay, and this is kind of a zero crossing analysis where 0 represents they're producing equal number of phis and equal number of cries. And postnatal day is on this axis, so they produce a lot of cries, and then they jump up and produce a lot of phis. Okay? And then this is across 10 different infants, a population plot that's showing the transition. Now, a lot of people would say, what's the big deal? This is just maturation. But really, that's not very satisfactory, because maturation is just, it's not like it's immune to experience, right? You have the experience not only of the infant producing its own vocalization, which is an experience, but in this case, and I'll show you, there's experience from other members of its family group. Okay, so how are we going to look at that? So on this plot, which is the population plot, you see these hatch marks. These represent the 10 individual marmosets, and when they transition in terms of postnatal day to producing more phis than cries. So that's their 0 crossing point. Okay? And now we're going to ask the question, how can we account for this variability? Okay? And what we did is we took these infants out, and we did this several times during their postnatal life. We took these individuals out and then had them spontaneously interact with their moms on one days and fathers on the other day. It turns out it doesn't matter if it's mother and father, so we just kind of collapsed across those data. We just asked, well, what is their interactions like with their vocal interactions like with their parents? Okay, and can we use that to predict the rate at which they transition from going to cries to phis? Okay, and remarkably, this is what the data look like. Okay, and here's why it's remarkable. Basically, on this axis is what was the proportion of contingent responses that are coming within 3 to 5 seconds from the parent upon hearing a mature sounding phi call from the infant. And then what you're looking at here is days that they transition from the cry to the phi. Okay, so there's a strong correlation between the parent giving a contingent response and the rate at which they'll make this transition. Okay, so to put it another way, if you're getting more contingent responses, if you're producing a fee and your mom produces a fee in response more often, you are going to more quickly develop your vocalizations in terms of that cry-fee transition. And its contingency seems to be important because if you just look at the total number of parent fee calls in that session, it has no correlation with the rate of that vocal development. Okay? And the final kind of vocal development data that I want to show you has to do with turn taking. So that was the previous example is a demonstration of a spectral tuning that seems to be influenced by parental interactions. This is a demonstration that that vocal turn taking behavior, that ability to wait 3 to 5 seconds before producing your own call, that actually has to develop postnatally as well. So when they're born, they're unable to control themselves and they just interrupt all the time. But over postnatal, a few postnatal weeks, then they develop that ability. And we're looking currently at the same relationship. Is the number of contingent interactions with the parent something that's predictive of how soon they will learn that turn taking?
How many years does it take for academics to mature on that?
I think they mature and then they lose it somehow. And then, you know, this is just another example where we can use the model to kind of simulate the development of that efferent copy connection and recapitulate the developmental curve. I'll skip this and go to the conclusions, which may be provocative. Okay? So what does this tell us about, what do Marmosets tell us about human vocal cooperation? Here's what I would like to say. Or here's what I'm going to say, and I hope you like it.
Do we have to listen?
So I think vocal turn taking is the scaffold for the evolution of more sophisticated communication abilities. So I'm not arguing that what marmosets, the content of what marmosets exchange, or even how they may be modulating their behavior is particularly impressive. What I think is impressive is that they're coordinating their call exchanges. They seem to facilitate trying to get the other person to hear that exchange. And without that, you really couldn't establish any other more complicated means of communicating, whether that's syntax, semantics, or whatever you choose to focus on. The similarities between marmosets and human turn-taking is an example of convergent evolution, at least at the behavioral level. And I want to talk about what may be going on at the neural level in just a moment. More generally, I think volubility and prosociality is an alternative mechanism that can lead to vocal cooperation. And in this mechanism, there is no need for a gestural root for this capacity and no need for a big brain. And that self-monitoring may be essential for vocal cooperation, and the circuitry may be shaped by postnatal interactive experience. Okay, so I'm going to close with an idea about convergent evolution and homology. So what I would like to argue, because I'm trying to promote this model system, so like anyone who's promoting a model system, you want to say, oh, it's just like humans, okay? I'm going to be perfectly blunt about that. But I think it could be interesting beyond attempts to get funding.
Oh, no, come on.
And here's why I think it's interesting. So it's convergent evolution at the level of behavior because we know if you look at I have an idea more comparative data are needed, but it's certainly the case that there are very few cooperative breeders in the evolutionary tree of primates, for example, within species of old world monkeys and certainly in chimpanzees. Okay? And so I think this cooperative vocal behavior kind of comes out of this prosociality that comes out of cooperative breeding. So it seems to be an example of convergent evolution at behavior level because there's no species in between that seem to exhibit this behavior, as far as we know. What I'd like to argue is that even though it's convergent evolution at the behavior level, it could still be mediated by homologous circuits in the brain. And I'm going to protect myself by using Leia's data to make the argument. So the reason why I think you can have homologous circuits comes from one of my favorite studies, which is by Padberg et al. From Leah's lab, where they're showing that in a distantly related New World monkey that basically develops the manual abilities that Old World monkeys like macaques and humans have, a precision grip, that once if they acquire this ability, which probably requires changes in hand morphology and the adoption of new behaviors, they actually kind of recapitulate the same area involved in that grip behavior, okay? An area that's absent in other New World monkeys. So in fact, you get this, there's a convergent evolution of behavior that basically leads to the development of the same circuitry, at least at the level of identifying an area. And so the conclusion is the range of cortical organizations that can emerge in primates is constrained, and those that emerge are the result of highly conserved developmental mechanisms. So I would argue that perhaps the same processes is occurring for this cooperative vocal communication, that once you get two systems that are cooperatively breeding, there's not a whole array of possibilities for the circuits that underlie those abilities, and that maybe the marmosets and humans could be using homologous circuits. And then I think evidence that kind of speaks to that directly or tangentially, depending on your sympathies, is also related to the stuff that Danielle presented, where males are carrying this circuit for caring for infant, but are typically not using it. And it's the modulation through experience and maybe hormones and other factors that can then activate that circuit to do what is essentially a radically new behavior. And maybe something like that is similarly going on in this vocal cooperation. So I'll just end here with basically addressing, well, where are we going with all this? Well, what we're currently collecting data in the lab is looking at respiratory EMG activity, its relationship to neural activity, and laryngeal tension, how that's developing, and then how that all relates to neurophysiology. And our neurophysiology is focusing on anterior cingulate and the auditory cortex. And our hypotheses are driven by the model that I presented here, and then another model which I didn't have time for, which is a biomechanical model of how infants produce those cries that eventually turn into fecal. So I'll end there, and thank you very much for your attention and questions.
Now we have more questions.
If you got the time, I got the time.
Is this on? Can I start?
Yeah.
I'm not sure using the term homologous circuits in these two lines is really the proper designation, because if you look at trichromatic vision, it evolved in the ancestors of Old World monkeys and in one branch of New World monkeys, but it evolved independently. So it's not the same. They're not homologous in that sense. It's independent evolution of a very similar sort of way of doing vision.
But if you get, so I'm not completely familiar with the trichromatic vision, but do their visual structures then adopt the same color-related properties that an Old World monkey has?
So the whole system hasn't been investigated enough to know to how similar they are. But the real key thing is to get gene duplication and then a third cone in there as a result of that.
Yeah, I understand what you're saying, but and I kind of I go back and forth because I think, well, how you can just keep switching the levels at which you want to define homology. So if I get to the same, if I have the same behavior that uses the same circuit, does how the circuit get there? Is that what's important for defining homology? If it gets there in a different way, does that mean it's not homologous? The circuit's connecting A, B to C in both cases.
I think it depends on what kind of question you're trying to answer. I think there are all kinds of interesting cases. So I'm sort of sympathetic both to what you're saying and to what John's saying. So the color thing, if I understand it correctly, is an interesting case where there really are independent roots. Of course, they're not fully independent. So they're all starting with the basic photoreceptor, and then there are branches, and the same kind of thing happen. Sometimes, like, I mean, there's the Richard Dawkins' famous thing about the eye developed 40 times independently isn't really true. The PAX6 is in all of these eyes across a wide domain, and so it's partly about the grain level that you're looking at, and partly about the question that you're trying to answer. So are you trying to answer a question about possible plausible roots? I like the quote from Leah's work about constraint. Are you trying to figure out what the constraints are? Are you trying to figure out what the molecular mechanisms are? I don't think there's a simple answer to, hey, you can use the words homology and convergence and so forth this way or that way, short of having a question that you're trying to frame.
Yeah, so I get your point. I think to answer your question, what we want to look at is the development of that circuit in terms of first connectivity, and then, depending on what we see, modulation of that circuit.
So you don't have the data yet to...
And I'm looking for that good neuroanatomist.
But so you don't have the data yet to distinguish between, say, there are common functional constraints that are leading to common places versus there's some common molecular precursors that get spelled out in different ways, in different contexts.
I need collaborators.
And let me go back 3 slides or so. The one that had phrased the scaffold. If you can. So I was, next, there. I was very sympathetic to almost everything there except for one word, and I wanted to ask you about that one word. So vocal turn taking is the scaffold for evolution.
Oh, sure. Yeah,
so why not a scaffold? I mean, like, why do you think this is a privilege, or do you think that it's a privileged piece of scaffold?
It should be an A. I agree. I have a whole other talk that argues a different line. So.
I was wondering, is there any evidence that maternal neglect or separation from the mother would interfere with this transition that you're showing between the crying and the fee cry? Or if there's any alterations maybe in the juvenile period, if they're separated from their sort of pack? That would interfere with that?
The stuff that the data that we have where they make the transition is much prior to the juvenile period. To answer your question, we do not have data, and we haven't done a deprivation-like experiment, but what we're doing currently is to do kind of an enhancement where we can use the playback to generate statistically more contingent responses. So we take the infant out, and it's interacting with the playback engine that's producing its mom's calls, and just giving way more contingent interaction than it would normally get. And our prediction is that would accelerate the development of that transition.
Hold on. Gary's getting his interview at 2:30, so we absolutely have to stop. You just ask him your question up there on the way to lunch. Okay, guys, you have to go to lunch. We're really behind schedule.
Thank you.